Good afternoon to all the NEET aspirants 2026. This is your Prasanna Ma'am from NEET World. So yes, in today's session we are going to exercise all the advanced set of questionnaire from anatomy of flowering plants. So yesterday we have dealt with all the NEET previous year questions from this chapter. Before that we have drawn out a clear analysis on the questionnaire pattern from this chapter, where we have looked into most of the multi-statement based questions, assertion reason questions, and match the following questions from this chapter. As such, we have designed few set of questionnaire. Let's get started. So this first question, which is directly from your NCERT. Okay. Good afternoon, Tanvir. Continue with the questionnaire, beta. So yes, in this question, which is directly from your NCERT diagram, okay, NCERT diagram, which is very important, that is shoot apical meristem, right? So here we have to look into the labelings properly. We know, unlike the root apical meristem, which is present at the which is present at the terminal region in the root. See, if you have to see the region of the root, where is root apical meristem present? At the terminal, subterminal or subapical region. But shoot apical meristem is present in the terminal location over here. So this region is what? Shoot apical meristem. B is what? Shoot apical meristem. And this is the leaf primordia. A will be what? Leaf primordia, which gives rise to leaves. Okay. And at the axil, okay, at the axil, axillary bud will be there. This axillary bud will be developing either into a shoot or a lateral branch. Okay, this will be what? Axillary bud. Axillary bud, which is at the axil. And these two are what? Differentiating vascular bundles or differentiating vascular tissue. This is what? Vascular tissue. Vascular tissue. Fine, right, beta? So look here, what will be the correct labeling? Tanvir, yes, you are right, Nana. B is the right answer. That is, A is what? Leaf primordium, B is shoot apical meristem, C is axillary bud, D is differentiating vascular tissue. Option B is the right answer. Next question. This, if you have to see, statement based question, beta. Try to answer this, Tanvir, and other students as well. So here, read the following statement and find out the incorrect statement. Whenever statement based question is asked, you have to look into whether in the question he is asking you to identify the correct set of statements or incorrect set of statements. Once after looking into that, underline over here and then look into the options carefully. So here if you see option A, there are structural similarities and variations or differences in the external morphology and internal structure of larger living organisms in both plants and animals. Yes, of course, there are variations or differences seen, correct? Next, plants have cells as basic unit. Yes, plants will have cells as basic unit, which is organized into tissues, and in turn the tissues are organized into organs. This we have learnt in cellular organization. Option C you see, beta. Very good, Tanvir, it is option C only. A tissue is a group of cells having common origin, usually performing different functions. What is the actual definition of tissue? Tissue is a group of cells which has common origin and similar in function. Tissue, the cells which are present in tissue can be structurally similar or dissimilar, but then they have to have a common origin and functionally they should be similar. They should not have different functions, functions should be similar. Structural dissimilarity is accepted. Okay, so here the incorrect statement is option C, but then we will also look into the option D. Axillary buds are present in the axils of the leaves and are capable of forming a branch or a flower. True, right? So here what is the incorrect statement? Option C. And then look at the next question, ma. Yeah. Assertion reason type of question. Okay. So both apical meristem and intercalary meristems are primary meristems, right? So both apical and intercalary meristems are primary meristems which help in the formation of primary plant body. And if you see in the reason, both these meristems appear early in life of a plant and help in formation of primary plant body. True, right? So assertion apical meristem and intercalary meristems are primary meristems, and what is the function of this primary meristems is clearly explained in the reason. Okay. So, assertion and reason are true, and reason is the correct explanation of assertion. So, here A and R are true and reason is the correct explanation of assertion. Option A is the right answer. Next question you look into beta. Yeah. So, here if you see, which of the following characteristics is not found in parenchyma? What is parenchyma? Parenchyma is a simple permanent tissue. There are few characteristics which are given for all the simple permanent tissues: parenchyma, collenchyma and sclerenchyma. So, here the question is about parenchyma. Good. Good, Tanvir. Look at this question, ma. It forms a major component within the organs. Yes, plant body is abundantly occupied with parenchyma only. Second statement you see, their walls are thick and made up of cellulose. Their cell walls are made up of cellulose, but then their cell walls are not thick, their cell walls are thin. Thin cell wall is present. Thick cell walls are not there, their cell walls are thin and they are made up of cellulose. Okay. So, option B will be the incorrect statement. They may either be closely packed or have intercellular spaces. Intercellular spaces are present in the parenchyma. The parenchyma performs various functions like photosynthesis, storage and secretion, correct. So, here which of the following characteristic is not found in parenchyma is the question asked. Which of the following is not found, beta? Option B will be the right answer over here. Okay. Next question you see. Yeah. So, here find out the mismatch pair. Mismatch pair we have to identify, and the question is about the xylem tissues. Okay. All the tissues which are present in the xylem, the question is about that. Here we have tracheids, vessels, xylem fibers and xylem parenchyma. Tracheids, if you see, tracheids are elongated, they are tube like cells with thick and lignified cell walls because their cell wall layers are made up of suberin, sclerenchymatous tissue. These are dead and without protoplast. So, they are dead. The statement is right only. In vessels, these are tube like structures made up of many cells. Okay. In tracheids, one tube is equal to one cell, but in vessels one tube is equal to many cells. Okay. Each with lignified walls and they have large central cavity. They are also devoid of protoplasm. True only. Xylem fibers, highly thickened walls and obliterated central lumens. Yes, the central lumen is very small. Okay, central lumen is very small. Xylem parenchyma, living and thick walled cellulosic cells. They store food materials in the form of resins, latex and mucilage. So, here you can see the wrong statement, that is xylem parenchyma only. Look at the statement once again, ma. They are living and thick walled cellulosic cells, they are saying. It is not thick walled, cellulosic cells are thin walled and living. All the rest of the three, they have what? Thick and lignified cell walls. Okay. Whereas xylem parenchyma, thin and cellulosic cell wall. So, here option D will be the right answer, that is the mismatched pair. Okay. Good. Next question you look into. Yeah. Which of the following statement is true? True statement he is asking. So, anytime statement based question, first identify whether he is asking you the right statement or wrong statement and just underline it over here, then you look into the options. In monocot plants, collenchyma occurs below the epidermis. Wrong. In monocot plants, what occurs below the epidermis? Sclerenchymatous hypodermis will be there. Sclerenchymatous hypodermis is present in monocot stem, not collenchymatous. Collenchymatous hypodermis is observed in dicot stem. Sclerenchyma is dead, thus without protoplast. Okay, sclerenchyma is dead and it does not have protoplast. True only. Xylem parenchyma is living, thin walled, lignified on cell walls. Wrong. It does not have living, it is living but it does not have thick lignified cell walls because it is having thin cellulosic cell walls. Here you see companion cells are specialized sclerenchymatous cells. Wrong. Companion cells are not sclerenchymatous cells, they are living cells only of phloem tissue. So, which is the only correct statement you can observe, beta? Option B is the right answer over here. That is the correct statement. Next you see, Assertion and reason. Okay, assertion and reason. Xylem vessel is long cylindrical tube like structure made up of many cells, each with lignified walls. Yes, xylem vessel is long and cylindrical. It is made up of many cells. Each cell has thick lignified cell walls. Next, reason, if you see, presence of vessels is a characteristic feature of gymnosperms. Wrong. In gymnosperms there are no vessels. There are no vessels in gymnosperms. In gymnosperms vessels are absent. So, here what you have to go through, ma, assertion is true and reason is false. Okay, assertion is true and reason is false. In that case, which option will be the right answer? Assertion is true. Yeah, the assertion is true and reason is false. Option C will be the right answer. Next question you look into. Yeah. This is about vascular tissues of angiosperms. Vascular tissues of angiosperms differs from those in gymnosperms. Look at the options keenly. In angiosperms and gymnosperms, once let us look into the xylem tissue, xylem and phloem tissues. So, vascular tissues include both xylem and phloem tissues. First, let us look into the angiosperms. Okay, angiosperms. Angiosperms, here you see it has what? Tracheids, vessels, xylem fibers, xylem parenchyma. Xylem fibers and xylem parenchyma. Tracheids, these are dead, vessels dead, xylem fibers dead, xylem parenchyma is living. All the 4 tissues are present in angiosperms. Whereas in gymnosperms, only tracheids will be present, xylem fibers will be present, vessels are absent, xylem parenchyma is also absent in gymnosperms. That is about the xylem tissue in the vascular bundles. Let us look into the phloem tissue also, both in angiosperms and gymnosperms. In angiosperms, if you have to take, phloem tissue is composed of sieve tubes, companion cells, phloem fibers and phloem parenchyma. Phloem fibers and phloem parenchyma. Okay. So, here in phloem tissue, if you have to see, sieve tubes, companion cells, phloem fibers and phloem parenchyma is present in angiosperms. In gymnosperms, sieve tubes are not present, whereas in gymnosperms, sieve cells are present. Okay. Sieve cells. Companion cells are absent in gymnosperms. In the place of companion cells, in gymnosperms you have albuminous cells. It has albuminous cells. Phloem fibers are present in gymnosperms. Phloem parenchyma is absent in gymnosperms. Clear, right beta? Clear with vascular tissues presence and absence both in angiosperms and gymnosperms. Now it will be easy for you to answer this question. Look at it. Presence of vessels in gymnosperms, wrong. Presence of vessels in xylem. So, the question is vascular tissues of angiosperms differ from those of gymnosperms. In angiosperms have the presence of vessels in the xylem, presence of well developed sieve tubes in phloem, true, presence of companion cells in the phloem. So, all the above statements are true only. Okay. Next question you see, vascular bundles in monocotyledons are considered closed because. So, now we have to explain the vascular bundles which are closed, which is the most apt statement for this. So, look at here carefully, a bundle sheath surrounds each bundle. Yes, true. In monocotyledonous leaf, bundle sheath is surrounding each vascular bundle. Cambium will be absent, but here we have to explain about only closed vascular bundles, right. Closed vascular bundle directly means that cambium is absent. Okay. Cambium is absent, which is not aiding in the process of stellar secondary growth. Okay. With the absence of cambium, there will not be stellar secondary growth. Stellar secondary growth is absent. Okay. Stellar secondary growth is absent. There are no vessels with perforations. No vessels will be there with perforations, and that is nowhere related to closed vascular bundle. Xylem is surrounded all around by phloem. No. Okay, closed vascular bundle just means that cambium is absent and there is no secondary growth that is taking place. Clear, right? Next, look at here. Given figure P and Q represent the stomatal apparatus of dicot and monocot leaves respectively. Select the option which correctly labels A, B and C. A, B and C. First you have to have a clear idea about stomatal apparatus. So, look at here carefully, ma. Stomatal apparatus will have what? Stoma or stomata. You can call it stomatal pore, stoma or stomatal pore plus guard cells plus subsidiary cells. So, these three cells together they form what? Stomatal apparatus. Now, you look at it carefully both in dicots and monocots. So, the dumbbell shaped guard cells indicate this is a dicot leaf. Okay, this dumbbell shaped guard cells is indicating this is a dicot leaf, and here bean shaped guard cells is indicating this as dicot leaf. Dumbbell shaped guard cells indicates this as monocot leaf, monocot. Fine. So, here in dicot leaf it has what? Bean shaped guard cells, and in monocot it has dumbbell shaped guard cells. At the center stomatal pore is present. So, stomatal pore, A is what? Stoma or stomatal pore, and C is what? B is what? Guard cells. And those specialized cells surrounding the guard cells over here, these are what? Subsidiary cells. So, see here will be subsidiary cells. So, here stomata, stomatal pore or stoma, guard cells and subsidiary cells. So, you look at here, A is stoma, B is guard cells, C will be subsidiary cells. In this case, what will be the right answer, Vita? Option D will be the right answer. Next question you look into. Yeah. So, this one. This question you see, Casparian stripes. The question is about Casparian stripes are the bands of thickenings present on dash walls of endodermis. So, Casparian stripes, which are the bands of thickenings, these are present in both radial and tangential walls, of endodermis in the root, both dicot root and monocot root. Endodermis is the last layer of the cortex, last cortical layer. So, this endodermis is having what? Radial and tangential thickenings of suberin. Suberin depositions in both radial and tangential walls. So, here it is option A and B, both A and B are right. Option D is the right answer. Next, monocot root. Monocot root you look into, cambium ring vascular bundles 2 to 4. Pith is large and well developed. Yes, pith is large and well developed. Next, radial vascular bundle. So, 2 to 4 vascular bundles will not be present in the monocot root. Diarch or tetrarch condition of vascular bundles is observed in the dicot root. In monocot root you will see polyarch vascular bundles which are more than 4. So, here option A goes wrong with monocot root. B you see beta, radial vascular bundles, polyarch xylem, no secondary growth. Yes, no secondary growth and polyarch xylem is observed, and then usually more than 6 xylem bundles. Pith is small and inconspicuous. Conjunctive tissue, pith will be broad and large, not small and inconspicuous. Small and inconspicuous pith you will observe in dicot root, not in monocot root. So, option C goes wrong here, and of course, D, B and C are not correct with respect to monocot root. So, here we have to go with option B. Option B is the right answer. Next question. Look at here. Yeah. What is correct about the monocot stem? Monocot stem, what is correct? Hypodermis is sclerenchymatous, vascular bundles are closed, phloem parenchyma is absent. True, right, with respect to monocot stem. And then hypodermis is sclerenchymatous, vascular bundles are open. No, vascular bundles are closed because there is no secondary growth in the monocot stem. Secondary growth is observed only in the dicot stem. And hypodermis is cholenchymatous. No, cholenchymatous hypodermis is observed in dicot stem. Hypodermis is sclerenchymatous, vascular bundles are closed, phloem parenchyma is absent in monocot stem, not present. So, in this case what option goes right, beta? Option A is the right answer. Okay. Next question. Yeah. Find out the incorrect statement about dorsiventral leaf. Dorsiventral leaf or dicot leaf is one and the same. Okay, dorsiventral leaf. Here you have to know what is the adaxial epidermis and abaxial epidermis. Adaxial epidermis is the dorsal epidermis or the upper epidermis. Abaxial is ventral or lower epidermis. Do remember that. And in a dorsiventral leaf, more stomata are present in the ventral surface, that is abaxial epidermis, and less stomata are present on the dorsal surface or adaxial epidermis to prevent the process of transpiration. So, here you see the adaxial epidermis generally bears more stomata than abaxial epidermis. This is incorrect because abaxial epidermis will have more stomata than adaxial. Here you see option B, the tissue between upper and lower epidermis is called as mesophyll, which is made up of parenchyma. True. The veins vary in thickness in the reticulate venation in the dicot leaf. Yes, the leaves will vary in thickness. If it is parallel venation, all veins are of same thickness. This is observed in dicot and parallel venation is observed in monocot leaf. And here if you see option D, the vascular bundles are surrounded by thick layer, that is bundle sheath cells. True, right? So, here what is the incorrect statement, ma? With respect to dorsiventral leaf, it is option A. Option A will be the right answer. Next question you look into. Yeah. Match column 1 and column 2 and select the correct option. Okay, we have to select the right option. Steel, if you see, all the tissues are inner to the endodermis. Yes, all the tissues inner to the endodermis are stellar region, wherein stellar region includes what, ma? Stellar region or stele includes pericycle, pith or medulla, and vascular bundles. Okay. So, it has pericycle, pith or medulla and vascular bundles. You can also consider medullary rays. Okay. So, here if you observe stele, all tissues inner to the endodermis, which is option 4. Next, endodermis. Endodermis is the innermost layer of the cortex, that is option 1, because cortex is made up of what? Hypodermis, general cortex, and the last layer of the cortex is endodermis. Okay. Next, Casparian strips. Casparian strips are what? Depositions in the radial and tangential walls of the endodermis in dicot and monocot root, and those depositions are what? Made up of suberin. Those depositions are suberin, which is option 2. Next, bark. Bark are all tissues outside to the vascular cambium. Okay, outside to vascular cambium, which includes primary phloem, secondary phloem, and periderm. Periderm means what? It has phellum, that is cork tissue, phellogen, cork cambium, and phelloderm, secondary cortex. All these tissues comes under what? Bark only, which is option 3. So, you look at the options carefully, beta. A is 4 and B is 1 and C is 3, A is 4, B is 2. Endodermis is the innermost layer of the cortex, B is 1. So, let me write the options. A is 4, all the tissues inner to the endodermis. B is 1, innermost layer of the cortex. C, Casparian stripes. Casparian stripes are made up of what? Suberin. C is 2. and D will be 3. Option A is the right answer. Okay. Next question. So, yes, this question is about the vascular bundle arrangement. Identify the type of vascular bundles as shown in the figure and select the incorrect statement regarding this. Okay. So, we have to identify the vascular bundle. Vascular bundles are radially arranged over here, radial vascular bundles. Okay. Radial arrangement of vascular bundles. Radial arrangement. Okay. Whereas in stem, if you see, it has conjoint arrangement where xylem and phloem are at the same radius. Can you see here, ma? Xylem and phloem are at the same radius. This mode of arrangement is observed in dicot and monocot stem. Can you see? Xylem is towards the inner side close to the pith and phloem is towards the periphery. When xylem and phloem are at the same radius, you call it as conjoint vascular bundle arrangement, whereas this is the radial vascular bundle arrangement where xylem and phloem are at the different radius. So, now with respect to radial vascular bundle arrangement, which is seen in the roots both in dicot and monocot root, we have to identify the right answer. So, A figure represents radial vascular bundles in which xylem and phloem occurs in the form of separate bundles. True. Xylem bundles and phloem bundles occur at different radii. Correct. These are the characteristics of monocot and dicot leaves. Wrong. So, incorrect statement we have to identify. These are not the characteristics of monocot and dicot leaf. In monocot and dicot leaves, they have conjoint closed vascular bundles. These are the characteristic. Radial vascular bundle arrangement is characteristic for monocot and dicot root, not leaf. So, here we have to go with option C as incorrect statement is asked to be identified. Okay. So, now let us move to the last question of today's session. Last question of today's session, which is diagram based question. Look at it carefully, ma. So, recognize the figure and find out the correct option he is saying. So, here upper epidermis or dorsal epidermis is what? Adaxial or dorsal epidermis, where the stomata will be less, stomatal distribution will be less. Whereas D is what? Abaxial or ventral surface of the epidermis. Abaxial or ventral surface of epidermis. Next, B, if you see, mesophyll is differentiated into palisade parenchyma and spongy parenchyma. So, B is what? Palisade parenchyma, which is below the dorsal surface or which is below the adaxial surface. Palisade parenchyma. Fine. And C is what? Spongy parenchyma. Spongy parenchyma. Fine. And then here if you see, this is what? Vascular bundles, where F is phloem and E is xylem. Fine. Now, you look at the options carefully. A is not abaxial side, A is just a second here, A is adaxial or dorsal epidermis and B is palisade parenchyma, C is spongy parenchyma, D is abaxial surface, F is xylem and E is phloem. Just a second, a small correction, F will be xylem, yeah, E will be phloem, right ma. So, this will be the labelings, and once again I would like to tell you all the diagrams which are present in the anatomy of flowering plants are important, be it in the initial pages of this chapter to identify the root apical meristem and shoot apical meristem, and then when it comes to the comparison between dicot root, monocot root, dicot stem, monocot stem, dicot leaf, monocot leaf, and then tissue systems as well, and then vascular bundles, both xylem tissues and phloem tissues, all the labelings are important. Okay. So, look at the diagrams with extra focus and extra interest when you are going through this. chapter. So that's all for today's session, ma. Let's exercise NEET previous year questions from another chapter of NCERT 11, which I will decide tomorrow. So tomorrow we will have again a NEET previous year question session. That's all for today. Thank you.